Time course infections of HeLa cells using Vaccinia virus begin by setting up the viral infections, infecting the cells, and then harvesting the RNA from the infected cells at several time points post-infection. This RNA can later be amplified for hybridization to microarrays for analysis of gene expression.
Hi, I'm Judy Yan from the laboratory of Kate Rubens at the Whitehead Institute for Biomedical Research. Today we will show you a procedure for a time course infection with the Vaccinia virus in mammalian cells and the subsequent RNA extraction from the infected cells at different time points post-infection. This technique is a beginning step in a larger protocol aimed to extract and amplify RNA in order to assess virus gene expression during infection, which we will complete in 2 other Jove videos. So let's get started.
To set up for the infection, grow HeLa cells in flasks and wait until the cells are approximately 80% confluent. Next, be sure to prepare enough viral growth medium for the experiment using regular DMEM with 2% FBS and no added antibiotics. The infection can be performed with either a crude stock of Vaccinia virus with a known titer or sucrose purified virus with a known titer if you're concerned about host gene expression. If you are using sucrose purified virus, skip to the next session. If you are using a crude Vaccinia virus stock, sonicate an aliquot of the virus just before infection in a cup sonicator with a bath of mostly ice and a little water. Sonicate it around 30 W for 20 seconds. Vortex the tube and repeat the sonication 3 times, adding more ice if needed to keep the cup packed with ice and chilled. Be sure to vortex in between each sonication step. If you do not possess a cup sonicator, mix an equal volume of the crude virus stock in 0.25 mg per milliliter trypsin. Vortex vigorously. Incubate the virus stock trypsin mix in a 37 degrees Celsius water bath for 30 minutes. Vortex at 5 to 10 minute intervals. To infect cells, first calculate the amount of viral particles needed to infect the monolayer at the desired multiplicity of infection, or MOI. Typically, a high MOI between 5 and 10 is used for infection time courses. Next, add the desired amount of sucrose purified virus or trypsinized crude virus to 37 degrees Celsius viral growth media and mix well. You should use enough media to just cover the bottom of the flask. 10 milliliters of media would be enough for a T175 flask. Remove the media from the cells and rinse with room temperature PBS. Add the virus or viral growth media to each flask. For a 0-hour mock time point, add the viral growth media only with no virus added. Swirl gently and tilt the plates. Incubate at 37 degrees Celsius in a 5% CO2 incubator for 1 hour. Be sure to tilt and swirl the plates every 15 minutes to spread the virus uniformly and keep the cells moist. After incubation, remove the media containing the virus and rinse 3 times with room temperature PBS. Add the maximum amount of viral growth medium to each flask. For example, 30 milliliters of media for a T175 flask. You can start counting this as your 0-hour time point. At the desired time point post-infection, check the cells under a microscope and note any cytopathic effects, or CPE. Remove the media and rinse the cells with 30 milliliters of room temperature PBS. Next, add 15 milliliters of trypsin to the cells and incubate for 2 to 5 minutes at 37 degrees Celsius. Check under a microscope for cell detachment and tap the flask gently to dislodge cells. Transfer the cells with a sterile serological pipette into a 50 milliliter conical tube. Wash the flask with an equal volume of cell growth media and add the media to the trypsinized cells in the conical tube. Centrifuge the cells at 500 Gs for 5 minutes at room temperature. After centrifugation, remove the trypsin media from the cell pellet. Resuspend the cell pellet in either trizol reagent or the lysis buffer from your desired RNA isolation kit. If you are using trizol, divide the sample into 1 milliliter aliquots and 1.5 milliliter labeled Eppendorf tubes and freeze at -80 degrees Celsius. Repeat for all time points, harvesting 1 flask per time point. At this point, your sample should be resuspended in trizol reagent and ready to be processed. Add 200 microliters of the BCP phase separation reagent for every 1 milliliter of trizol in each tube. BCP, or 1-bromo-3-chloropropane, is used in place of phenol to reduce genomic DNA contamination. You may need to transfer the sample to a larger tube if you are starting out with a large amount of trizol. Vortex or shake vigorously and incubate at room temperature for 2 to 3 minutes. After incubation, centrifuge the sample at 12,000 Gs for 15 minutes. After centrifugation, an aqueous phase should be visible as a clear layer on top. Transfer the aqueous phase to a fresh tube. You should be recovering approximately 600 microliters for every milliliter of trizol you started out with. Do a second phenol chloroform extraction, this time using 500 microliters of chloroform per 1 milliliter trizol instead of BCP. The second chloroform extraction is used to remove any traces of organic solvent from the extraction, as even trace amounts can inhibit subsequent amplification steps. Vortex or shake vigorously, then incubate at room temperature for 2 to 3 minutes. Centrifuge the sample at 12,000 Gs for 15 minutes. After centrifugation, transfer the aqueous phase to a fresh tube. Remember, the aqueous phase is the clear layer on top. Next, add 20 micrograms of linear acrylamide to each sample. The linear acrylamide acts as a carrier and helps to precipitate the RNA. Add 500 microliters of isopropanol per 1 milliliter of trizol you started out with to each sample and mix well. Incubate the samples at room temperature for 10 minutes. After incubation, centrifuge in a microcentrifuge at maximum speed for 10 to 15 minutes. The RNA pellet will be visible at the bottom of the tube after centrifugation. Very carefully remove the supernatant from the tube, either with a vacuum aspirator or manually by pipetter. Do not disturb the pellet. Wash the pellet with 1 milliliter of 70% ethanol and centrifuge at top speed for 5 to 7 minutes. Very carefully, remove the ethanol from the tube either with a vacuum aspirator or manually by pipetter. Discard the supernatant. Check that the pellet is visible at the bottom of the tube. Repeat this wash step. After the supernatant is discarded, mark where the pellet is on the tube. Air dry the pellet for no longer than 5 minutes. Do not over dry or the RNA will be difficult to reconstitute. Next, if DNase treatment is planned, resuspend the pellet in 17 microliters of nuclease-free water. If DNase treatment is not planned, resuspend in 20 microliters. For DNase treatment, use the QIAGEN RNase-free DNase set. Add 2 microliter buffer RDD and 1 microliter reconstituted RNase-free DNase 1 to the resuspended RNA. Mix gently. Incubate at 37 degrees Celsius for 30 minutes. Next, add 2 microliters of 2.5 millimolar EDTA and incubate at 65 degrees Celsius for 5 minutes to inactivate the DNase. Do not exceed the inactivation time or temperature, as longer times and higher temperatures can cause degradation of the RNA. Now you have completed the DNase treatment. Check the RNA concentration by spectrophotometer. Traces of phenol or BCP can be detected as a spike at 270 nanometers, beyond the standard peak at 260 nanometers, when measuring the absorbance of total RNA after extraction. If such contamination appears, re-extract the RNA using a filter or column-based RNA extraction method before proceeding with amplification. You can store the RNA sample at -80 degrees Celsius. You can check the RNA quality if you desire by using an Agilent bioanalyzer or by running the sample on a denaturing gel and checking the OD readings. On the bioanalyzer, good RNA quality is indicated by the RNA integrity number, or RIN. The RIN is based on a numbering system from 1 to 10, with 1 being the most degraded profile and 10 being the most intact.
We've just shown you how to perform a time course infection with the Vaccinia virus and how to extract the RNA from infected cells at various time points post-infection. When doing this procedure, it's important to remember that there are several critical steps in setting up a synchronous Vaccinia infection. First, careful sonication or trypsinization of the virus is important to disaggregate virus particles, since Vaccinia is highly prone to aggregating, which would prevent an even infection of cells. Second, in order to achieve a synchronous infection, a high MOI of greater than 2 should be used to ensure each cell is infected. A mixture of infected and uninfected cells will lead to multiple rounds of infection, heterogeneous mixtures of time points, and asynchronous viral and host transcriptional responses. Third, the infection should be carried out in minimal amounts of media to allow maximum absorption of the virus onto the cells. In addition, regular shaking of the flask or culture dishes every 10 minutes enables distribution of the virus across the flask and ensures that the cells do not dry out. So that's it. Thanks for watching and good luck with your experiments. Be sure to watch the other videos in this series, where we show the procedure for linear amplification of RNA samples and the preparation and cleanup of fluorescently labeled RNA.